Plant versus intruder, or how plant cells defend against fungal pathogens. Crop production is undoubtedly crucial for our lives. Therefore, we strive to protect the plants from various kinds of pathogens, like bacteria, viruses, microscopic fungi, or pests infecting plant tissues, which thrive at their host's expense. Every year, a significant amount of crop is destroyed, leading to financial losses and thus rising costs of food. Currently, various chemicals are employed to prevent or fight the infections, but they are often costly and in some cases damaging to the environment. Plants, however, can neutralize most of the pathogens themselves, despite having nothing like the immune system we find in animals. But how does that work then? How do plants beat their intruders? What weapons do they use? Such questions have fascinated scientists all around the world. Indeed, the answers might contribute significantly to our constant struggle with crop diseases. Now we are on the leaf surface of a dicote plant, consisting of epidermal cells bearing hair-like extensions called trichomes, and stomata through which the plant allows gases to enter its organs. Plus, air and waterborne pathogenic microorganisms are all around. For example, here comes a microscopic fungal spore. After germinating, its hyphae seek to get inside the plant in the easiest possible way, either through stomata or an open wound. Some of them are even able to penetrate the epidermal cells directly. At a closer look, we see that the plant employs a sophisticated passive defense, a protective shield of a water repellent or hydrophobic wax layer called cuticle, and underneath an armor with a sturdy cell wall made of polysaccharids, mainly cellulose. Cellulose is extremely strong, and for many organisms it is mechanically and chemically impenetrable. The intrinsic integrity of the cell is drawn out by a thin and flexible plasma membrane. Germinating fungal spores form a swollen ending called aprosorium, which they utilize as an adhesive sucker wherein the pressure reaches up to 17 MPa. This is a pressure 25 times higher than that in usual bus tires. The aprosorium, therefore, works like a sort of hydraulic battering ram. The fungal aprosorium also releases exosomes, vesicles containing enzymes that decompose the cell wall of the plant. In general, the cell wall of fungi constitutes of chitin, a wholly different kind of polysaccharide that is recognized as a foreign structure by receptor proteins, such as SIRC1, found in the plasma membrane of plants. The fungus counteracts by releasing more exosomes that fuse with the membrane, injecting the cell with its effectors that break down the plant defensive proteins to make the plant cell let the infection in, unrecognized and defenseless. This is an epidermal cell. It contains all the organelles except the chloroplast. You can see the nucleus, the endoplasmic reticulum, the mitochondrion, and the enormously spacious vacuole that is filling most of the cell. Vesicles carrying various kinds of cargo are moving along the cytoskeleton, pulled by molecular motors. All of these organelles and structures cooperate to combat the pathogen. The environment of the cytocell is acidified by introduction of hydronium cations being released from the mitochondrion and the vacuole. Further, calcium ions that are essential for signalization pour out from the mitochondrion and the endoplasmic reticulum. At the same time, the vacuole also releases salicylic acid, It signals the attack of an intruder, stimulates production of oxygen radicals, triggers transcription and translation of defensive proteins, and also becomes a messenger itself, alerting the surrounding tissue. Therefore, it is one of the phytohormones that propagates stress signals into adjacent cells through tubular connections known as plasmodesmata. Another plant agent works in cooperation with salicylic acid. It is the enzyme NADPH oxidase, which oxidizes NADPH forming superoxide, an extremely reactive oxygen radical. Such radicals oxidize proteins, membrane phospholipids, and nucleic acids. It serves as an urgent general alarm signal in the cell. However, damaged proteins must immediately be decomposed to prevent their accumulation and to prevent the cell from collapsing. That is the role of the proteasome, or more commonly known as the cellular waste grinder, wherein proteins are broken down into recyclable amino acids. Meanwhile, signalization is still occurring on the cell's surface, where SIRC1 receptor has detected molecules of fungal chitin. The signal subsequently proceeds further into the cell by means of the MAP kinase cascade, which activates a transcription factor that in turn commences transcription of its respective gene-encoding chitinase, a chitin-cleaving enzyme. Chitinase is synthesized on the rough endoplasmic reticulum and then transported in vesicles on the cell's exterior, hindering fungal growth in its surroundings, subverting its attack. Another defense protein is cellulose synthase, a transmembrane protein which moves along microtubules immediately underneath the plasma membrane, polymerizing single glucose molecules from cytosol into long chains of cellulose released outside of the cell. Thereby, the cell wall is reinforced. At first, the cell wall thickens at the site of aprosorium, and then also all over the cell's surface. The plant cell has another crucial mechanism in its defensive arsenal, used in case of a very dangerous specialized intruder attack. NBS-LRR, a control protein, serves as a cell's toughest policeman, guarding some of the other plant proteins. If any of the associated proteins are broken down by fungal effectors, NBS-LRR alerts its respective transcription factors, allowing the final phase of defense to begin. Thus, a cell that struggles with a specialized pathogen commits cellular suicide. In other words, the cell perishes together with the aggressor within. Before that, the plant cell isolates itself from its surrounding tissue. It thickens the cell wall and blocks plasmodesmata by polysaccharides. The level of oxygen radical increases, whereas the cytosol pH decreases. Microtubules are being broken down. The genetic information in the nucleus is shredded, and the nucleus, together with the endoplasmic reticulum, collapses into a myriad of vesicles. A cataclysmic culmination of the programmed cell death follows. The vacuole membrane fuses with the plasma membrane, inundating the fungus with its acidic contents. The cell has sacrificed itself for the sake of the plant as a whole. Even though it may not seem so at first glance, plants are tough rivals for pathogens. During the battle, they are even willing to sacrifice regiments, or let's say individual cells, for the final victory in the war. Cell death has a major role in other important processes besides the protection against pathogens. For example, in cell development, the structural components of conductive tissues, tracheas, and tracheids are foraged from the remains of cells dying. Leaf shed in autumn is enabled by programmed cell death too. Each multicellular organism gains its shape not only by cell division and growth, but also by coordinated cell death. For the cell, death is an integral part of life.